Cell cycle and cancer is the focus of this review video, and just to get us started off, or to give us an overview of what will be included in the video, we're going to be talking about the overall or the major reasons a cell would divide, and the most basic would be just to produce more cells in an organism called a eukaryote. And a eukaryote is an organism like yourself. It's a more complex organism, usually a multicellular organism, although some single-celled organisms that are eukaryotic do exist. And then we have our prokaryotic cells, and those are single-celled organisms. The best example of that would be something like a bacterial cell. It's a very simple cell. It's a much smaller cell, has a lot less going on inside of the cell membrane compared to a eukaryotic cell. And the division process is also quite different from prokaryote to eukaryote. And the overall purpose is very different as well. The overall purpose in a prokaryote would be to really reproduce and make another organism, although that organism would be a genetically identical cell in prokaryotes. Whereas in eukaryotes, this process of cell division we'll talk about here, in particular it's called mitosis, is really to make more cells, for instance when cells are damaged, or to grow or develop from young to an adult. And we'll be talking about the stages of the cell cycle, in particular for the eukaryote. And again, that cell cycle in the eukaryote is called mitosis. If we go on to our next slide here, we look at the division process for the prokaryotes. It's called binary fission, and the overall process is shown here. Now for our prokaryotic friends, their DNA, which we'll talk more specifically about in coming chapters, is one single circle. It's a single circle, and they have one chromosome. That is their genetic material, and it's housed somewhere inside of the cell. It's really not encased in any kind of a particular compartment, such as a nucleus. They don't have a nucleus. And this single circle is copied, as you can see here in the diagram, and the cell divides. So it's a pretty straightforward, simple process here in the prokaryotes. Now with eukaryotes, it's a little bit more complex because these are more complex cells. And just to give us an idea of some of the difference here in the eukaryotic cells, instead of a single circle, we have linear strips of DNA called the chromosomes. So there are many of these linear strips, as opposed to in the prokaryotic cell we just have that one single circle of DNA, which is their one chromosome. So that's a big difference there. And also in the eukaryotic cell there are many more genes, because there is just a lot more genetic material comparatively. And just to give us some terminology here, the chromosome is made of individual units, or put together by individual units of DNA called genes. Each of these different colored boxes here represents, for instance, a gene, and each of those boxes would have a particular genetic sequence or DNA sequence on it. We'll talk a lot more about DNA in particular and the structure and function of DNA in incoming chapters. So look forward to that. All right, we'll move on. So we'll talk more about this division process. So our chromosomes, represented here as that purple looking structure, linear structure at the very top of the diagram, they replicate during the first phase of the cell cycle, and when they replicate, they are joined together in the center by that second figure down by a centromere. So it's a connection point between the two sister chromatids. So we have what was one of the old chromosomes and then a brand new chromosome, but when they're connected they're called sister chromatids. And then during the division process they separate, and once they are separated they are now individual chromosomes, and they will be distributed to the two daughter cells, the two cells that result from this division process. Alright, So in this next slide, I have this beautiful, beautiful picture, electron micrograph of sister chromatids connected by the centromere. And that is just lovely. It's just highly, highly coiled DNA, and oh, beautiful. Okay, so the cell cycle. We have, this is represented here in more of a pie chart kind of view. And it really does show that the phase called interphase is the longest phase of the cell cycle. And it's actually divided into 3 subphases, G1, S, and G2. I'm not going to have you, know what goes on in particular in these subphases, but just to know that the cell does grow in interphase. It gets larger. This DNA is synthesized, in particular in the S subphase. So we make our sister chromatids in that particular phase. And the cell's really preparing. It's preparing for the division, the mitosis. Okay, so this is really the longest phase, and then we have the mitotic phases, which are 2 through 5 listed here. And as far as these phases are concerned, as far as what you need to know, you're going to want to know the order of these phases, interphase through telophase, and the major events. All the major events are outlined in the slides and in the textbook. And then you're also going to want to recognize the real pictures of real cells that I have here in this PowerPoint and also in the PowerPoint for this chapter. So again, be able to recognize those cells in particular. You will see these cells again, and I'll show you those as we move along. So interphase, again, the cell's growing, the chromosomes are condensing and duplicating as well. The nuclear envelope begins to break down. And the picture here is of a cheek cell, and you can see that there's really not much going on in that nucleus. You really can't see any individual chromosomes yet. They're beginning to condense, but they're really not visible as linear structures. And then we move on to prophase, and you actually can see the chromosomes as linear structures inside that nucleus. And the nuclear envelope is further breaking down. You have your centrosomes, which are forming this early spindle, which are going to be used later in mitosis. And then we have our picture here representing prophase. You can see the individual chromosomes in the center there as individual structures, linear strips. Metaphase, all the chromosomes are lined up single file in the middle of the cell, attached to the spindle. And that's our representative picture here of metaphase with our real cell. And then anaphase, the sisters separate, and now again each of these chromosomes, once separated from its sister, is an individual chromosome. The cell begins to elongate, kind of take on an oval-like shape as those sisters move to opposite poles. And that's our representative picture. And then telophase, we start to have the chromosomes uncoil. The nuclear envelope begins to reform in each of the daughter cells, and we see this cleavage furrow forming between the 2 new cells. And the mitotic spindle disappears, dismantles, and mitosis is complete. So this is a representative of really telophase going into something called cytokinesis, where we actually see the 2 daughter cells as 2 complete structures. And the daughter cells are usually a little smaller than the cells surrounding them. That's how you can tell they've really just gone through mitosis. Okay, and again, cytokinesis here, really right along with telophase. They kind of happen at the same time. And cyto means cytoplasm, really, or stands for cytoplasm, and kinesis means to cut. Okay, and then looking more into this idea of the cell cycle and how sometimes it can go awry and not listen to the signals the cell is getting from the outside to divide or not to divide. And sometimes cell division can get out of control, and that's really what cancer, the disease cancer, truly is just one cell that has gone crazy, and it is dividing out of control, and all of that excess cell division forms a tumor. There are different tumor types. Over here we see a benign tumor. The tumor is a real nice tight ball of cells shown here in pink, and it is in one structure, and it really hasn't diffused through that structure very much. And this is really a good prognosis if you find it in this stage, because it can easily be removed, or oftentimes the organ is just removed as a precautionary measure. And now it can develop, if left undetected, into a malignant tumor, shown in the second diagram from the left. And in the malignant, the cells really aren't adhering to each other as well any longer. They're not in that nice tight ball of cells. They're more diffuse through several tissues in the organ, and it's a poor prognosis. And usually the entire organ will be removed. And sometimes people will receive chemotherapy or radiation depending on the extent of the tumor. And then finally, if the malignant tumor is not detected, it can become metastatic, meaning that the cells are even less adherent to one another, and they actually break off from the original tumor, and they travel through the blood vessels or the lymph vessels to other parts of the body, and they create metastases or tumors in other parts of the body. Oftentimes the brain or the lung or the bone are common sites. And this is a very poor prognosis. Radiation chemotherapy are oftentimes used to combat or extend life in this kind of situation. So I hope that has helped you better understand cell cycle. Have any questions? Please let me know.